Alright, so we're going to spend a few minutes now talking about the structure of the plasma membrane and pretty quickly get into some cell transport processes like diffusion and osmosis and a few other things that we need to talk about to kind of set the stage for dealing with how cells interact with their environment when it comes to getting things and getting things out. So the first thing we'll talk about, like I said, is the structure of the cell membrane, which we've talked about a bit before, but we're going to get a bit more in depth into it now. So as far as functions of the plasma membrane, and you'll hear me use the term plasma membrane and cell membrane kind of interchangeably. Technically, plasma membrane is a bit more specific because you could use the term cell membrane for really any membrane inside of a cell, and there are tons of things inside the cell that have membranes, right? So, like the nucleus, the ER, the Golgi, lots of different kinds of membranes in the cell, but a plasma membrane is specific to the membrane that surrounds the cell. And the job of this structure is to, like it says, isolate the cell's contents from the environment. It's a barrier to movement. It allows some things to come in, other things to go out, other things not so much. So it's kind of like almost like a bouncer that's very selective in who it lets in and who it lets out of the cell. So we have this exchange component as well. Some things can go right through a phospholipid bilayer. Other things have to have some sort of help. And like I said a few minutes ago, that's something we're going to delve into here in the latter parts of the chapter. We have a degree of cell communication. So a lot of times plasma membranes in multicellular organisms are going to have receptors on the outside that will allow them to receive messages from other cells elsewhere in the body. So we have a communication aspect here as well. We have attachment points. So, if you notice, if you grab your skin and pull, hopefully it doesn't just come off. The cells in your body are anchored together through different types of attachment points. And that's actually the very last thing we're going to talk about in this chapter is different types of cell-to-cell attachments that we find throughout the cells in the body. We also have in many cells, especially prokaryotic cells, enzymatic activity in the plasma membrane. So there will actually be enzymes built into the membrane itself, which help to facilitate different types of chemical reactions. And so for a prokaryotic cell that doesn't really have a whole lot of internal structure and lots of organelles and things like that, they will actually use their membrane to do just about everything functionally that cell has to do. Right? So there's no internal organelles. They have to use their membrane. Now, one of the things I really want you to look at on this slide and focus on and think about is the fact that structure and support are not part of what the plasma membrane does to support a cell. The plasma membrane is somewhat fluid. It's liquidy. It does not have a really firm, durable physical structure. It's really good at allowing things to come in and go out, but what it doesn't do is to provide a shape to the cell. All right? You can't have a fatty layer provide a physical, consistent shape to a cell. So, for cells that have a sort of definitive physical shape, that shape does not come from the plasma membrane, but instead can come from things like a cell wall or can come from things like the cytoskeleton that we talked about at the end of the last chapter. So, physical structure, support, shape, those are not things the membrane does to support the cell. So the bulk of the plasma membrane, like we talked about previously, is made up of phospholipids. And if you remember back to our chapter on kind of the biologically important molecules, a phospholipid is made up of a polar part, which is the head, so to speak, if we pick out one of these phospholipids right here, this head is made up of a glycerol molecule and a phosphate group, which is polar. So it's hydrophilic, right? Now these tails are hydrocarbon chains. They don't necessarily have to be the same as each other, but they are made up of carbons and hydrogens in different degrees, sometimes with double bonds, sometimes with only single bonds, but the tails are the hydrophobic part of the phospholipid. So these phospholipids have a hydrophobic pair of tails, kind of on the back end here, and a hydrophilic phosphate glycerol part up here at the head end. And that's the part that helps to interact with water. So when you have a lot of phospholipids together in a watery environment, an aqueous environment, what they're going to do is to join up and form a bilayer with the hydrophobic tails pointing toward the inside and the hydrophilic heads pointing toward the outside of the cell and the cytoplasm of the cell accordingly, right? Because this is hydrophobic inside of here, this is hydrophilic, this is hydrophilic. So they're going to join up spontaneously and form a phospholipid bilayer, which is, of course, the main part of the barrier component of the plasma membrane. Right? So with the plasma membrane, because most of the internal structure is hydrophobic, that means that hydrophobic molecules will be able to pass through relatively easily back and forth across that membrane. Hydrophobic things can pass back and forth as long as they're not too big. They can go back and forth either out of the cell or into the cell all day long without any sort of protein help, without any sort of energy help. They just sort of do so through simple diffusion because they are hydrophobic, and most of the inside of the cell is also hydrophobic. Remember our rule about solubilities, like dissolves like. Hydrophobic passes through pretty easily, even though it's got to squeak past these hydrophilic heads. That's not too hard to do. This hydrophobic molecule can go back and forth. Now, on the other hand, hydrophilic molecules, even though they get along with these surface phospholipid heads here and these surface phospholipid heads up here, they cannot cross the hydrophobic tail part of the bilayer. So they're kind of confined either outside the cell or inside the cell, unless the cell has some sort of other help to get that molecule in or out. And we'll talk about that more here in a little bit. All right, so as far as the kinds of things that can use simple diffusion back and forth without any help at all, anything that's hydrophobic, so fats, oxygen gas, carbon dioxide, those things are all polar, and they can all pass back and forth relatively easily. On the other hand, hydrophilic things like ions, water, carbohydrates, amino acids, those things have a much more difficult time crossing a phospholipid bilayer directly, so they're going to need to have help through the use of transport proteins and sometimes through the use of energy as well. All right, so a good way to describe the phospholipid bilayer, we already used the term selectively permeable. That just means some things can go through it, other things can't, or if they do, they need help. The other really good way to think about the phospholipid bilayer is as a fluid mosaic. All right, so a mosaic in sort of artistic terms is where you take a bunch of little colored pieces of glass or stones or even paper and arrange them to form a picture made up of smaller things. The idea that it's fluid means that those little pieces that are arranged together are able to move back and forth. They're not attached to each other, so they can move back and forth relatively easily, and therefore, over time, even short periods of time, change the overall structure of the phospholipid bilayer. So a fluid mosaic means there's all these different kinds of molecules kind of moving back and forth around each other. They're not really anchored in place so much, and that gives the membrane a degree of fluidity. The two layers are held together in the center by hydrogen bonds. The hydrophobic tails do participate to some degree in hydrogen bonds, which help to keep the two layers anchored together, even though the individual phospholipids primarily are unanchored most of the time. So remember, hydrogen bonds form and break relatively quickly, relatively fast, fairly frequently. So even though one part of a bilayer might be anchored to another part for one moment in time, the next moment in time it may not be, and it might be sort of floating around at will if that hydrogen bond has been broken. So the bilayer is actually very fluid in its nature as well. So here's just a quick picture of what one author imagines a phospholipid bilayer to look like. Primarily, we have all these little dots and tails. Those are phospholipids. We have a whole lot of different kinds of proteins we're going to talk about here in a second. We have the cytoskeleton inside the cell. We have all kinds of other proteins outside the cell in the extracellular matrix. It's actually a very complicated environment, the surface of these cells, which is why we actually give the membrane its own separate chapter as opposed to covering it with the rest of the parts of the cell back in chapter 4. Now, as far as the main parts of the membrane kind of classified, we've already talked about phospholipids, right? So we talked about what they do. There's also cholesterol present in the phospholipid bilayer, which helps to give it a degree of fluidity that is correct most of the time. So you want the membrane to be somewhat fluid, but you don't want it to be too fluid. You want the kind of Goldilocks just right, kind of in the middle sort of a thing. And cholesterol molecules help to maintain the right level of fluidity regardless of temperature. Right? Make sure the membrane is the right bendiness or degree of liquidity all the time because of the cholesterol molecules present in the membrane. We also have what are called transmembrane proteins. So transmembrane means it goes across the entire membrane. So it goes from the outside of the cell all the way through the membrane to the inside of the cell. We also call those integral proteins. So the transport proteins we're going to see here in a few minutes are a good example of a type of transmembrane protein. We also have what are called peripheral proteins, or proteins that are just on one surface of the membrane, either out here on the outer surface, maybe in here on the inner surface, but they do not span the entire membrane itself. Right? So those, a lot of times, enzymes would be a good example of a type of membrane that's a peripheral protein that's only on the surface of the cell, either inside or outside surface of the cell. And then finally, we also have our surface markers, and these things can be glycoproteins, so a protein with a carbohydrate side group. They can be glycolipids, which again is a carbohydrate group with a lipid side group. And so those are some of the things that, for example, recognition proteins a lot of times are glycoproteins, and they're a way for a cell to signal that it belongs, right? So cells in your body have different kinds of external markers than cells in somebody else's body. And that's one way that our immune system knows who to attack and who not to attack in our body, is by recognizing the protein signatures on the outside of the cells. Well, a lot of times those protein signatures are glycoproteins, and they help to show the other parts of the body that a certain cell belongs, as long as it has the right kind of protein structure on the outside. All right, so we've talked about this already. The phospholipids not attached to each other most of the time. We talked about briefly that the fact that the two phospholipid tails are not necessarily identical. One phospholipid tail could be saturated, and for that reason solid at room temperature. Other fatty acid tails could be unsaturated and therefore liquid at room temperature. So the degree of bendiness of the phospholipid tails is based on the presence or absence of carbon-to-carbon double bonds, and whether the lipid is a saturated fat or an unsaturated fat. We talked about the fact that hydrophobic things can pass through pretty easily. So fatty acids, steroid hormones. If you ever take Anatomy and Physiology 2, at Lakeland at least, the way we teach it is that the endocrine system is in the second semester of A&P. And one of the things we look at really early on is the difference between how protein hormones and lipid hormones actually affect their target cells. And it turns out that lipid hormones affect their target cells by passing through the membrane. And the only way that lipid-based hormones or steroid hormones can pass through the membrane is because they are hydrophobic, and the membrane, of course, is also hydrophobic. So they're able to pass through the membrane, whereas protein-based hormones, which are hydrophilic, they have to find receptors that are on the outside of the cell because they can't pass through the membrane directly. We also have the limitation of hydrophilic molecules from moving through the membrane. So again, ions, amino acids, carbs, most proteins, those guys are not going to be able to pass through the membrane directly because the membrane is hydrophobic. These molecules are hydrophilic, so they're going to have some sort of help. Now, the other two things that we need to think about just really briefly when it comes to the physiology of our bodies is oxygen and carbon dioxide. And you might think that both oxygen and carbon dioxide should be polar because they have oxygen present. It turns out they're not. And so the reasons we could get into, if you have specific questions, we can talk about why this is. But both oxygen and carbon dioxide are hydrophobic molecules. And as such, they are able to pass through the membrane directly without any help because they're hydrophobic, and of course the membrane is too. And then we have cholesterol maintaining fluidity, like we talked about before, making sure it's the right level of bendiness all the time, regardless of temperature. All right, now the last thing we're going to talk about in this part of the chapter is the different types of proteins that we find present in a plasma membrane. So the first type of protein that we'll look at is a transport protein. All right, so transport proteins are just what it sounds like. They are proteins that are going to allow things in or allow things to go out of a cell. So they're like the doorway for hydrophilic molecules to enter or exit a cell, because otherwise, of course, those molecules can't do it on their own because they're hydrophobic. These transport proteins allow things to come in or go out of a cell at will. We also have enzymatic proteins. Enzymatic proteins are just enzymes, and we're going to talk a whole lot more about enzymes in the next chapter, but an enzyme for our purposes right now in this chapter, really all you need to know is an enzyme is a protein that helps to speed up the rate of a chemical reaction. And as we said before, sometimes enzymatic proteins are built into the plasma membrane, and they're going to allow the reactions to go faster or slower, usually faster, at body temperature to allow them to create the products they need to create. We also have receptor proteins. So in this picture over here, we have a green molecule. This could be like a protein hormone, like insulin, for example. Insulin comes along. If it finds a receptor on a cell that it fits, it plugs into the receptor, and then that triggers a series of other reactions inside the cell, which is represented by this, to allow the cell to respond. All right, so protein-based hormones, in large part, in vast majority of cases, are going to affect their target cells by plugging into these receptors and therefore activating some sort of response inside the cell. We have our recognition proteins, like we talked about before. Recognition proteins are those proteins on the outside of a cell that a cell can use to identify itself as belonging to the body. Right? So your immune system won't attack cells that have the right receptor proteins, hopefully. And so they're very important for that reason. A lot of times these can also be glycolipids as well, or glycoproteins as well, but a lot of times they can just be proteins, almost like an ID badge or a fingerprint on the outside of a cell to signify that that cell belongs and should not be attacked. And then finally we have attachment proteins, and we'll talk about this more in the last section of the chapter, but basically there's three types of attachment points between cells. There are anchoring junctions, there are tight junctions, and there are what are called desmosomes. And those are going to allow cells to connect, but sometimes have other properties as well. And in most cases, those attachment points are created by membrane proteins too. All right, so a few other just illustrations of this. This blue protein in this picture actually is a receptor protein. So we have our pink molecule plugging into the blue molecule. So again, this could be like a protein-based hormone that's plugging into a receptor. And when it does that, the receptor turns on, reactions happen inside the cell, and the cell responds accordingly. All right, so this is a very common situation for protein-based hormones to affect their target cells this way. The main key feature of that is that the target cell has to have a receptor if it's going to respond. If a target cell doesn't have a receptor, then if the hormone comes by, doesn't see a receptor, it's going to go right on its way and go find a cell that does have the receptor. So that's a really critical way that hormones very specifically affect only certain cells, is because the receptors for those proteins are only on the target cells. I think we're actually going to skip that slide. And from there, we're going to move into the different types of transport across the membrane. So I'm actually going to cut this section off here. It's actually a good place to take a break and start up with our next big topic in this chapter when we talk about diffusion and osmosis and facilitated transport and active transport and bulk transport. So we'll cut that off here, and we'll start up again in just a second with the next section.